Thank you very much, and thank you for the invitation to come speak. So I'm going to talk about the BioCompute standard today, which is a descriptive standard that I think fits very nicely. So I'm going to shift gears a little bit and talk about standards development, which is fun. So I'll tell you a little bit about the standard and kind of the origin story so that you know where it came from and why it's a thing. And then I'll talk about the BioCompute Nextflow integration. Okay, so the roots for this idea go back to 2012, back when NCBI was putting together their data model, and they were trying to come up with a way to represent and organize all of this different but very related data. And it ultimately became known as the Bio Project, and they've got this very nice model here. But during those conversations, it was suggested, wouldn't it be nice if we could also have some way of representing all of the descriptive metadata in a way that's computable. And that was still kind of a nebulous idea. So although there was some support for it at the time, it was not, I think, well articulated enough and did not ultimately make it into the Bio Project. But the seed for that idea was planted. So the idea was that it was hoped that you could have some way of recording how all the data in a data repository or academic journal or regulatory filing or something like that had been generated instead of maybe one line that says X software was used, and that's it, because that's a little bit difficult to make any informed scientific decision on, right? And so this is very much akin to a wet lab experiment. You couldn't just give somebody an image and say, this is it, we're done. You'd have to explain what was the animal, what was the tissue type, what was the antibody, and give some context, like what were you trying to do and what was the purpose and things like that. And so we have felt this, I think, in academia, at least in reviewing articles, I think very powerfully, but probably more concretely, it was felt more concretely in terms of measurable impact, it was felt in regulatory submissions to the FDA. And so what typically happens is, in fact, I'm told almost always what happens, is an industry sponsor will submit something to the FDA for review, and whatever that computational component is, the reviewer will kick it back with need for clarification. And so they'll then have to figure out what wasn't clear and review it. This is not a situation where you can just sort of sit someone down next to you at your computer and sort of explain what you did. It has to go through all the regulatory filings, draws everything out by months, and quite literally millions of dollars. And this is all just to get the FDA enough information to make a decision. So it's incredibly inefficient, really starting to, I think, cause frustration on both sides. And one of the most, poignant examples that I heard was when an industry sponsor submitted something and the reviewer kicked it back, and they said, we don't know, the postdoc that ran that analysis left 8 months ago. And so there's not really anything that they can do with that, unfortunately, which is kind of tragic. And this is despite the fact that it was perfectly re-executable. It was just that nobody understood the workflow well enough to be able to explain it and answer questions about it. And that's maybe not uncommon. Maybe there's a postdoc or a grad student or someone in your organization who's really kind of poured their heart and soul into this analysis, and they know all the details. And so if they leave, if it's not very well documented, then all that tribal knowledge really goes with them. So one potential solution to this was for reviewers to just sort of publish their own recommendations. This is not official guidance because that has a very specific meaning in the world of regulatory review. But this essentially just said, if you're going to submit this type of computational work to us, here's my recommendations for doing it. Here's what I'd like you to do. Here's how I'd like it to be represented. In this case, it was next generation sequencing, an NGS reviewer. And this was a fantastic step in the right direction. The problem was that this is not standardized across the whole agency. It's very much, any individual reviewer that wants to do this will do this. It's also very trial and error. So if something doesn't work, then they'll go back and they'll review it and try again. But it was a great step in the right direction. And so in 2014, what was then called the Genomics Working Group at FDA met and tried to sort of tackle this problem and begin to articulate what the issue was. And so they produced a document after lots of meetings that is summarized in these bullet points here. So any solution to this problem should be human readable. It should be machine readable, meaning we can compute on these structured predefined fields. It should contain enough information to understand the computational pipelines, interpret that information. That's really the core of the idea. And then finally, it should be immutable. So one of these records would be sort of like a snapshot in time of all the bioinformatic resources that were used, who the person was, the input data, et cetera. it would be a specific record of exactly what was done. So that idea that was first proposed in 2012 was pitched at that meeting, and it was accepted, and which by then was being called BioCompute, and so off they went. And I don't have any really cool NASA analogies to go along with my timeline. That was a really cool idea, but other cool things were happening during this time too. So after several workshops and couple rounds of funding, several collaborations and testing, several publications, 2 of which are annotated on this timeline, some testing, we finally ended up, and so we had input from hundreds of people across dozens of organizations. So lots of people in one way or another, made their voices heard as we were trying to hash out what this would look like. And so this is the model that we ultimately ended up with. It's written in JSON, and we just sort of abstract a pipeline away and take the conceptually related information and put it into these top-level categories called domains. So there's 8 domains. 5 of them are required, 3 of them are optional. And the idea here was that we wanted something that would be flexible enough to support any pipeline, but still rigid enough to provide some sort of computable structure for all the metadata. And ideally something that's not overly burdensome, because the whole idea here is that we're trying to speed up and make communication more efficient. We just need to kind of set some guidelines on what do we need to know and how do we want to represent that. So this is the data model that we ended up with. It has very strong data provenance. It has very strong user attribution and very strong descriptive metadata. And so this is really kind of the niche where this sits. And so in 2020, it was officially standardized through the IEEE organization as 2791-2020. And so we all continue to call it BioCompute, even though that's not the official name anymore because it's much easier to pronounce. So the standard itself is through IEEE, which means that it's paywalled, but all of the schema files are in an open access repository on GitLab. And so the link is here if you'd like to go check those out. If you do, you can start with that top-level file called 2791object.json. That'll reference all of the other files. And so within months of publication of the standard, it was accepted at the FDA, which is kind of like light speed at the FDA. Nothing happens that fast. In fact, to this day, it's still the only framework standard like this that has ever been adopted by the FDA's data standards catalog. So this was a really big win for us, and I think kind of signaled the need on both sides, how much people needed some framework for talking about computational pipelines. And so at that point, the focus kind of shifted towards building out an ecosystem of plugins and packages and tools and software and things to work with the standard and make it easier for people to author these BCOs, as they're called, BioCompute Object. So any pipeline that's written in a way that conforms to the standard is a BCO. So make it easier for people to read and write and represent all of these. And so that brings us to the NFProb plugin. So this is a really spectacular plugin. Looks like I am missing a single quote on one of those lines, but hopefully you get the idea. So the basic idea is it's very simple. You just enable it in your config file and give it the output file name, give it the path, and then this plugin will listen for all the workflow events that happen during the workflow's lifetime. So a task executes, a task completes, a file is published, all kinds of runtime metadata, and then a BCO is rendered. And thanks very, very much to Ben Sherman, who's the person who developed this plugin. So it's really fantastic. And so you'll get most of a BCO for free, which is step 4 there. There's still a few things left that we could potentially add in there. We may possibly add in some more fields in the config file. We may try to work with what's there, but in any case, the current state of it is really terrific. And so now you'll get this, anytime you run, for example, something from NFCore, or really any Nextflow pipeline. It doesn't have to be from NFCore, but any Nextflow pipeline, you can now get a specific record of that instance, essentially for free. And then in step 5, you can upload that to our portal if you would like to further annotate your pipeline, or you can annotate it manually. You don't have to use our pipeline, but you can go in and add some additional things. For example, some free text description. And so that's the basic idea. So as I said, we wanted to keep it simple. So the very simple model is intentionally straightforward. So who are you? What's the platform or the environment that you're working on? Is it cloud or your own laptop or whatever? What is the input and output files? What are the data transformation steps? And then finally provide some kind of high-level description around the entire thing to give some context and some background. So it's intentionally very, very straightforward, but it is extensible. So you can extend it out to a lot of things, and I think something like a workflow manager is a very natural extension for it. So this is our BioCompute portal. We do have our own builder here. This is a standalone builder, so if you're not working on a platform and you want to, it's just a form-based field, so it kind of walks you through which fields are required. So it's, if you want to manually build one, which I recommend if you've never done it before, just to kind of see what goes into a BCO. But from then on, it's probably going to be much easier to work with some of these tools that build it for you automatically. So we've got a lot of other resources up there, documentation, FAQs, things like that, some of the events that we have, the BioCompute community that's been heavily involved in this. We've also got a few different ways of viewing BCOs. This is a color-coded table. We're working on some other ones to try and, again, just make the whole thing an easier user experience for people, including FDA reviewers. We're getting input from them and from folks in the community. So please feel free to check those out and give us any feedback that you would like. We would love to hear your thoughts. And so that's it. I would very much like to thank, first and foremost, Raja Mazumdar and Vahan Simonyan. They were the two original architects in 2012 who brought this up at those NCBI meetings. Ben and Phil have been fantastic to work with, and also, I should have put on this slide, our mutual colleague, Kostis Karagiannis at Moderna, who kind of helped to broker this entire collaboration. But Ben put that entire thing together, and it was fantastic. It's just been wonderful working with them. Our own BCO team, several folks at the FDA, including Luis Centeno-Quintero and especially Mark Walderhog, and the BioCompute Regulatory Advisory Board, and of course our funding from the NIH and the FDA, and I'm happy to take questions.
Thanks, Jonathan. We have a question. Does BioCompute plug into or overlap with the workflow-related GA4GH standards?
That is a great question. So we had one representative from GA4GH that was part of our meetings. He, I think, ended up as the first author on the standards paper. We don't currently have any direct interlap. We're sort of, or overlap, or compatibility, I guess I should say. We are exploring that right now, and all I can say at this moment is that we kept that perspective in mind, thanks to his representation when we were building it, but there's no explicit plugin right now. One thing actually that I want to mention, I just realized, we're also in the middle of doing a pilot project right now with the FDA. We've got a few companies on board, and this is basically, now that the FDA has support for this standard, and there's tools for working with BCOs and things like that, we want to just sort of map out the logistics of all of this so that we know, like, there's questions around like, well, what piece of a pipeline would be represented, and do you want multiple BCOs that reference each other, or these kinds of questions. And then just the logistics of like, how do you actually transmit a BCO to the FDA? So we're organizing this pilot project to work out all of those details. We've got a few companies on board. We've got a few of the reviewers at the FDA on board. If you have any interest in participating in that, we would also be very happy to recruit you as well.
Thank you.
Sure.